Next is chapter 6, cell, the unit of life. First, the word cell comes from the Latin word celle, which means a small compartment. The word cell was first used by Robert Hooke, 1662. Therefore, the term cell is as old as 300 years. Now, Robert Hooke was used எப்போனா 1662. அதுக்கடுத்து டிஸ்கவரி Aristotle 3842 322 BC was the one who first recognized the animals and plants consist of organized structural units, but unable to explain what it was in 1660s. அதுக்கப்புறம் தான் தெரியும். ராபர்ட் ஹூக் observed something which looks like a honeycomb with a grand members of little boxes, which was later called as cell from the cork tissue. In 1665, he compiled his work as micrographic level. Later, Anton von Leeuwenhoek observed unicellular particles which he named as animalcules. Robert Brown, 1831 to 39, described the spherical body of the plant cell as nucleus. H.J. Dutroux, French scientist, was the first to give idea on cell theory. அதுக்கப்புறம் cell theory propose பண்ணது யாருன்னா மேத்தியாஸ் ஸ்கெலிடன், ஜெர்மன் botanist, and Theodor Swan, German zoologist, 1833. outline the basic feature of the cell theory கொடுத்தாங்க. அதுக்கப்புறம் எக்ஸ்பிளைன் பண்ணியிருந்தாங்கன்னா ருடால்ஃப் விர்ச்சோ, 1858, can explain the cell theory by adding a feature stating that all living cells arise from pre-existing, முன்னாடி இருந்த செல்லேருந்து தான் தோணுச்சு அப்படின்னு சொன்னார். லிவிங் செல்ஸ் பை செல் டிவிஷனில். அடுத்து மைக்ரோஸ்கோபி. எப்படி மைக்ரோஸ்கோப் வந்ததுக்கப்புறம் தான் செல்ஸ் எல்லாம். மைக்ரோஸ்கோப் இஸ் an inevitable instrument in studying the cell and subcellular structures. It offers scope in studying microscopic organisms. Therefore, it's named as microscope. Micros, small, scorpion, to see in Germany terminology. In the Greek terminology, common microscope was first invented பண்ணது யாருன்னா ஜட் ஜான்சன். Microscope basically works on the lens system and its properties of light and lens, such as reflection, magnification, பெருசாகிறது, and numerical aperture NA. The common light microscope, which has many lenses, are called common microscope. The microscope transmits visible light from sources to eye or camera through sample. Bright field microscope. The bright field microscope is the routinely used microscope in studying various aspects of cells. It allows light to pass directly through specimen and shows a well distinguished image from different portions of the specimen. The contrast can be increased by staining the specimen with a reagent that react with cells and tissue components of the object. The light rays are focused by condenser onto the specimen on a micro slide placed upon the adjustable platform. நீங்கள் ஆர்டினரியாக லேபில் பார்த்துருப்பீங்க, called stage. The light comes from the compact fluorescent lamp, CFL, or light emitting diode, LED. Then it passes through the two lens system, namely objective lens closer to the object and eyepiece, நம்ம கண்ணில் பார்க்கக்கூடியது. அதில் எத்தனை லென்ஸ் இருக்குன்னா 5x, 10x, 45x, மேக்னிஃபை பண்ணுங்க 100x. which can be rotated and fixed at a certain point to get a required magnification. It was on the principle of numerical aperture value and its own resolving power. The first magnification of the microscope is done by the objective lens, which is called primary magnification, and it's real inverted image. The second magnification of microscope is obtained through eyepiece lens called secondary magnification, and virtual and inverted image. Resolution power. The term resolving power or resolution refers to the ability of lens to show the detail of object lying between 2 points. It is the finest detail available from an object. It can be calculated using the following formula. என்ன அப்படின்னா resolution 0.61 lambda by NA. Lambda is wavelength of the light and NA is numerical aperture. Numerical aperture is it's an important optical constant associated with the optical lens, denoting the ability to resolve. Higher the NA value, greater will be the resolving power of the microscope. Magnification. The optical increase in size of an image is called magnification. It is calculated by the following formula. Magnification: size of image seen with the microscope by size of the image seen with the normal eye. Electron microscope. Electron microscope was first introduced by Ernest Ruska 1931 and developed by Pinning and Roer 1981. It is used to analyze the fine details of cell and organelle called ultrastructure. It's used to be beam of accelerated electrons as source of illumination, and therefore the resolving power is 1 lakh times greater than that of light microscope. The specimen to be viewed under electron microscope is dehydrated, water irukadu, ada dehydrated and impregnated with electron opaque chemicals like edulana gold or palladium. This is 1 mark for the withstand electron and also contrast of the image. There are 2 kinds of electron microscope, namely transmission paraval electron microscope TM, அதுக்கடுத்து scanning electron microscope SEM. TEM பார்த்தீங்கன்னா transmission electron microscope. This is the most commonly used electron microscope which provide 2-dimensional image. 1 mark. The components of the microscope are as follows: electron generating system இருக்கும், electron condenser இருக்கும், specimen objective, tube lens and projector. படம் உள்ளது இந்த டயக்ராமில் இங்கே electron microscope பார்க்குறோம். The beam of electron pass through the specimen to form an image on fluorescent screen. The magnification எவ்வளோ இருக்குன்னா is 1 to 3 lakhs times. Resolving power பார்த்தீங்கன்னா 2 to 10 Armstrong. It is used for studying detailed structure of virus, mycoplasma, cell organelles, etc. அதுக்கடுத்து scanning electron microscope. This is used to obtain 3 dimension. அங்கே 2 dimension பார்த்தா இது 3 dimension image and has a lower resolving power than TEM. In this electrons are focused by means of lens into a very fine point. The interaction of electrons with the specimen result in the release of different forms of radiation such as Auger electrons, secondary electrons, backscattered electrons from the surface of the specimen. These radiations are then captured by an appropriate detector, amplified and then imaged on fluorescent screen. The magnification அங்கே 1 lakh சொன்னால் இங்கே 2 lakhs times. Resolution is 5 to 20 nanometer.